to Hao Chen of Singleron Biotechnologies, who is going to introduce our first speaker in more detail. Over to you, Yuan.
Thank you very much, Richard. And it's a great pleasure to have everyone here today. We have wonderful speakers. And the first speaker, we would like to invite Dr. Hao Chen. He is at the moment at Institute of Reproductive Medicine in Nantong University. We have started collaboration from 2018. And Dr. Chen is also one of the early users and early adopters of our technology. And during these 3 years, we have produced quite a lot of single-cell data. And very interestingly, we had once a discussion together regarding how we could measure the nascent RNA during development. And this discussion leads to one of our newly released product, DynaScope. And Dr. Chen today will also introduce some unpublished data from this very exciting new methods. So with that, we would like to welcome Dr. Hao Chen on stage. Dr. Chen, it's yours.
I think his connection has just dropped off by the look of it. Maybe we should give him 30 seconds.
Yeah, seems to be a little bit of internet problem. Yes.
Oh.
Hi, Dr. Chen. Welcome. You're muted.
Yeah.
Now, perfect.
Yeah, now it's okay. My internet was broken.
Okay, now the stage is yours. Thank you.
Okay, so still I'm first, or
yes, yeah, you're ready to go now.
Okay, yeah, sure. Thank you very much, and thank you for your introduction, and Dr. Chen, and thank you for the invitation of Richard, and thanks for having me to introduce our recent work and some preliminary results. I'm going to present the spatial. Sorry, I can. Okay. The spatial-temporal landscape of mouse epididymal cells and the special mitochondrial-rich segments defined by large-scale single-cell RNA-seq, which had been published on Cell Discovery. As you may know, spermatozoa from the testes acquire their motility and fertilization ability when they transcend through the epididymis. It's a critical male sex organ that plays key roles in sperm transport, maturation, and storage. As shown in the slide, the mammalian epididymis is divided into 3 parts, named capt, corpus, and cauda. The middle figure is the HE section wheel of the different epididymal segments, which displays distinct morphologies. In general, luminal secretions from the capt and corpus are beneficial of the acquisition of sperm motility and fertilizing ability. On the other hand, cauda epididymal secretions maintain the condition for the storage of spermatozoa while preserving their fertility. The cartoon of the figure C shows the composition of the epididymal epithelium, which include principal cell, basal cell, clear narrow cell, and some immunocells so far. During the past decade, many studies were performed from single gene to omic study, both in mouse and human, in order to elucidate the role of the epididymis and its effect on sperm maturation. However, although advanced results were achieved in the segmental specific regulation, the mechanisms underlying the sperm maturation remains elusive. And a recent review by Sala appeared that we need to draw attention to recognize the distributed epididymal environment as a potential cause of male infertility. And with the technology development, single-cell RNA sequencing provide a novel scale to elucidate the potential regulation of the microenvironment at a single-cell level. Many organs have been analyzed in mammals, including the male and female reproductive organs, such as the testes and the ovary. And the table at the right panel has summarized analysis of the germline cell development from pre-implantation to the spermatogenesis. However, the detailed cell composition and their marker genes of the epididymis were largely unknown. With the question above, we performed the microfluidic-based single-cell RNA sequencing to analyze the cell divided from the capped corpse and the chorda from the mouse epididymis. The left photo displays the conventional method of epididymal segment region. On the other hand, we also analyze different developmental age, representing the first wave of spermatozoa. The time point is 42 days, and the subsequent wave of sperm maturation, the time point is 56 days, respectively. In total, we have the 40,625 cells were analyzed at the large-scale number level and underwent the further analysis. We first performed the cell quality control analysis for all the samples. After cell or gene selection, variation regression, and data normalization, and the multiple sample integration, the cells were visualized by UMAP and clustered into, you can see, 9 populations. And the epididymal cell type annotation was based on the known epididymal cells marker genes. And we collect from the previous published study. And the cell was labeled as figure in C. From this figure, we can see the C5 of this cluster is blood cells. In general, the blood cells will is excluded from the previous study. Therefore, we excluded the blood cells in the further analysis. And last, the cells were clustered into 9 populations, which include all known epididymal epithelial cells. And the specification of cell markers in cells was shown in the left heat maps. Consistent with a previous study, the percentage of principal cell was around 60% in the 3 epididymal regions. Intriguingly, the number of segmental differentially expressed genes, means DEGs, was consistently higher in the capped epididymal cells. It's well known that the expression of epididymal cells shows segmental specific or distal change the patterns in the epididymis. To further elucidate the segment specificity in the context of gene expression profile, we paired the DEGs in all cell types along the 3 segments, and the representative heat map was shown in the figures. The representative segmental specific genes in principal cells also elucidated. To clarify the subpopulation in principal cells, we employed a higher SNN clustering resolution, and 8 different subpopulations of the principal cells were revealed from the capital coda, which were defined as PRC1 to PRC8. And figure B, the top 10 DEGs were illustrated. Furthermore, the geo-enrichment analysis, which associated with the DEG in each cell cluster, geo-enrichment analysis, which associated with the DEGs in each cell cluster, manifested diverse roles of principal cells in regulating the epididymal microenvironments. Intriguingly, genes enrichment in the PRC7 and PRC8, the 2 small cell group, and were related to cilium organization and assembly, microtube-based movement, and the cilium-dependent cell motility, according to the analysis, suggests its role in epididymal cilium formation. Of note, these 2 clusters of principal cells were consistent with histologically described epididymal cells, with stereocilia accompanied the expression of actin. Therefore, we plotted the potential marker genes of PRC7 principal cell subsets and visualized them in the volume plots. The detailed functions of these cells were warranting to be investigated in further study. And similarly, other epididymal epithelial cells also can be clustered. In detail, the basal cell was able to subdivide it into 4 clusters. The DEG and geo analysis were also deciphered to show the divergent role of subcells of the basal cells. Again, the clear cells can be subclustered into 4 subpopulation too. Apart from the cell clustering, the cell-cell communication is a critical way to understand the epididymal microenvironment regulation. And with a package of CellTalk DB and single cell single R, we found that wide paracrine communications between each pair of cell type were discovered. Interestingly, non-epithelial cells such as endothelial cell and the macrophage or monocytes was able to affect epididymal epithelial cells, including principal cell, clear or narrow cells, and basal cells. And in particular, the potential pair of cells of communication while ligand receptor was deciphered. For example, the communication from the endothelial cells to from the endothelial cells to the principal cell and basal cell and clear cell. In addition, and the macrophage or monocyte were found to also can influence other cell clusters such as principal cells and basal cell. And these results indicated the complex cell-cell interacting among the epididymal epididymis. As we all know, the percentage of mitochondrial transcriptions is the key factors of cell quality. In general, the high level of mitochondrial genes suggested poor sample quality. However, Daniel compared mtDNA percentage of 165 tissues of mouse and human, which suggested some organs have higher mitochondrial transcripts, such as heart and kidney. Unfortunately, the epididymis was not included in their analysis. So unexpectedly, the consistently higher percentage of mitochondria transcripts was observed in the corpse and the cauda epididymis than in the capped. Based on the P42 epididymis results, an average of 7% of UMIs or MT mitochondrial transcripts in the capped segment, while some of corpse and cauda segment was around 15%. Such higher mitochondrial transcript percentage in corpse and cauda segments was reproducibly observed in two replicates of the P56 epididymis. And furthermore, you can see the mitochondrial transcript percentage in 4 epididymal cell populations or analysis, and the high mitochondrial percentage was observed in the corpse and the cauda segments, as well as in the global mitochondrial transcript analysis. To confirm more single-cell sequence observation, we use the mitochondrial tracker staining and immunofluorescence staining to examine the distribution of mitochondria in epididymal epithelial cells. Indeed, either the mitochondria specific marker, cytochrome C, or mitochondrial tracker staining shown a significantly higher mitochondrial number in the corpse and cauda than in the capped epididymis in both P42 and P56 mice. In addition, we also use the cytochrome C to staining the staining the epididymis. Here is the enlarged figure. In addition, we validated the mRNA expression level of selected mitochondrial genes by qPCR. In line with single-cell RNA sequencing results, the mitochondrial genes such as MTCO1, MTCYTB, cytochrome C, and MTND126 were highly expressed in the corpse and cauda compared to the capped epididymis. So in summary, our data provide the indispensable resources at a single-cell resolution for the comprehensive cell atlas and the spatial temporal profile of the epididymis. And importantly, our study find a high mitochondrial content in the epididymal epithelial cells of the corpse and the cauda compared to the capped epididymis. And our study has not only demonstrated the cell compositions and unique spatial temporal mitochondrial characteristics, but also shed light on the understanding of the normal epididymis functionalities at the cellular mechanism of epididymis diseases that cause male infertility. In the next section, I would like to share some of preliminary results on the cell atlas of dynamic transcript genes during spermatogenesis. As you may know, the single-cell RNA sequencing is fitting to identify the cellular identities and the cell type specific markers. However, current data of single-cell RNA sequencing is only the snapshot of the particular time point of organs. It cannot explore the dynamic of genes transcription. Interestingly, a novel method was developed in 2019, which is called scRNAseq, by using the biochemical nucleoside conventional. Similar with above study, Singularine developed the first commercial service for the detection of nascent single-cell RNA-seq named DynaScopes. And here's the workflow of the DynaScope. With the DynaScope technology, we investigated the dynamic transcriptions during spermatogenesis, and the left figure was diagrammed is a diagram of our experimental. And after sequencing, the quality control analysis was first evaluated to the evaluated the data quality. From the right figure, the number of feature RNA was over 20,000 on average, and the mitochondrial transcripts were under 5%, and there were no genes of blood cells were detected. And overall, this sequence data was good enough to further analysis. And the second step for the analysis was cell clustering. As you can see, the cells were able to be clustered in 9 different populations, and the marker genes were shown in Figure B. The percentage of each cell was calculated in the left panel of notes. The nascent RNA in each cell cluster was charted in the right picture. From the results, you can see the percentage of the nascent RNA in the stem cell spermatogonia or early spermatogonia cells, and the spermatogonia cells and the Sertoli cells was much higher than the late stage of the germ cells, and suggesting the limited transcriptive activity after mitosis. So we further investigated the potential involvement of the process of nascent RNA in the different cells, and for such as SSC and the spermatogonia, you can see the SCSA is mainly enrichment with the chromatin remodeling or cell proliferation, and the right panel, the spermatogonia, the most enrichment is cell respiration. And last, we examined the cell-cell communication between the different cell clusters, and found the most cell-cell interaction between mitochondria and Sertoli cells, and suggesting its important roles during spermatogenesis. And collectively, although we have the preliminary results for the nascent RNA during spermatogenesis, further analysis and experimental validation still should be performed. Okay, let me see. Yeah, and one more thing, I'd like to share the rigorous journal. I'm as a young editor of the innovation, and this journal covers all disciplines of science, and we welcome any interest to submit the paper. At the end of the presentation, I would like to give my sincere thanks to my former advisor and co-workers, and would also like to extend my thanks to my colleagues and the postgraduate students. And thanks for your attention. That's all.
Thank you very much, Dr. Chen. That was really, really interesting, especially these unpublished data.